Hi guys, we're going to talk about the microbiology topic 10, Macaque, oxidase, and gram-negative rods. So, let's talk first the family Enterobacteriaceae and their description naman. So, members are free living in nature and also found in the intestines of man and animals. Because of their habitat in man in the intestines, they are collectively called enterics, term also used for other bacteria that are found in the intestines. For example, Pseudomonas and anaerobic bacteria normally found in the intestines. In this lecture, it only pertains the family of Enterobacteriaceae. So let's talk about the clinically relevant members. So we have opportunistic pathogens, or also known as the coliforms. They are enterics that are part of the common flora on the gastrointestinal tract of man. However, if they are given the chance to invade other body sites, these are common cause of pneumonia, meningitis, septicemia, UTI, wound infection. And the example of these are the Escherichia, Klebsiella, Enterobacter, Serratia, Citrobacter, Proteus, Providentia, and Morganella. So we have also the overt pathogens. These are cause disease in gastrointestinal tract of man, not part of the normal flora, only found in the man at the time of infection. Their mode of transmission is fecal-oral route, except Yersinia pestis, which transmitted through a bite of an infected rat flea. So the example of the overt pathogen is the Salmonella, Shingilla, and Yersinia. So let's talk about the biochemical and morphological properties of the Enterobacteriaceae. So they are gram-negative Glycobacilli. They oxidize negative, which except for Pseudomonas species, a new member of the family, belong to another group before. Well, they grow well on Macaque agar and on using methylene blue agar or EMB. Both are differential media. Their ferment glucose is acid or acid and gas, which anaerobic is only acid is produced from glucose. Aerogenic if both acid and gas are produced. So they are facultatively anaerobic, and they are motile, which they describe as the peritrichous, except for Tatomela piticius, which is apolar flagellated. There are also be non-motile like Shingilla, which they are overt pathogen, and Klebsiella, which they are coliform. So they grow well on routine media like blood agar, culture agar, DHIB, and thioglycolate broth, just like the cocci. So the blood agar and culture agar have the same components. However, the culture agar is cooked after the addition of the blood. They are also asporogenous, which do not form spores. They reduce nitrate to nitrite except for Pleurohabdus, Xenohabdus, and some strains of Enterobacter and Yersinia. They are catalase positive except for Shingilla dysenteria type 1 and a species of Xenohabdus. So there are two types of plates of your blood agar. The first image shows pure culture of an enteric organism. Only one organism is growth. In the second image, you can see two types of colonies. The big ones are the colonies of an enteric bacterium, while the medium-sized colonies are the colonies of Staphylococci. You can see that the enterics will produce bigger colonies than the Staphylococci on blood agar. The Staphylococci are described as medium-sized colonies on blood agar, while the enterics are described as large colonies on blood agar. How about the Streptococci? Let's talk about that later. So, what's next? So, we're going to discuss the antigenic structure, which there is somatic antigens, which there are OAJ, which means that all enterics have this antigen found on the polysaccharide side chain of lipopolysaccharide or endotoxin components of outer membrane. The O antigen is H stable. Identified with specific O antiserum wherein we do agglutination test to detect the O antigen. Then we have the capsular antigen, which there are KAJ, VIAJ. This is designated as K antigen in Enterex except the Salmonella because capsulated Salmonella have the V antigen. Cover the O antigen because the capsular antigen is found in the capsule of the bacterium which surrounds the cell. So their heat label, it means that can be destroyed by boiling. We make a bacterial suspension of the colonies growing in the medium, then we place this in a boiling water bath for 10 minutes, which will destroy the capsular antigen and will expose the O antigen. Then you can repeat the test for O antigen using the specific O antiserum. So we have also the flagellar antigen, which they are designated as the HAJ. It is found in the chone that makes up the flagella. Their heat label can be destroyed by heating and can be destroyed with ethanol. Serotyping should be done using colonies from a non-sugar containing medium, for an example like blood agar, to prevent false positive results. Hence, you can use colonies growing on blood agar, culture agar, nutrients agar, or trypticase soy agar which do not contain sugar.